Hello everyone. In this video, I am going to discuss about nitrogen fixation. This nitrogen fixation is one of the most common and important topic for ICAR JRF exam plant sciences and as a part of general agriculture for all other subjects. So stick with me until the end of the video to have a clear-cut understanding about nitrogen fixation and also different microorganisms associated in this process. Okay, before talking about the nitrogen fixation directly, let us discuss about nitrogen first. So why this nitrogen is important for living organisms? So nitrogen is a major component of amino acids and nucleic acids in all the living organisms. So this is why the nitrogen is very important. So how this nitrogen is, we will just discuss like how this nitrogen is absorbed and utilized by the plants and animals. Before that, let us talk about the available forms, like in whichever form this nitrogen is available in the environment. So the nitrogen is majorly available in two different forms. One is organic and second one is inorganic form. So the organic form of nitrogen is again the amino acids and the nucleic acid. So the inorganic form of nitrogen is atmospheric nitrogen, that is N2, and ammonia or ammonium or in the nitrate form. So these are the inorganic form of nitrogen that are present in the environment. So in the environment, this nitrogen will be converted from one form to another form in a cyclic process, and there are several mechanisms which will aid this transformation. So the different mechanisms which will help in the transformation of nitrogen from one form to another forms are ammonification, nitrification, denitrification, and biological nitrogen fixation. So we will just discuss all of this mechanism one by one. So let us start with biological nitrogen fixation. So what is this biological nitrogen fixation? So before that, let me tell you about nitrogen fixation. So what does this mean? So the nitrogen fixation is nothing but the conversion of unavailable form of nitrogen to available form. As I told you, the nitrogen is available in the different forms in the environment, but the plants cannot absorb this nitrogen in all the different forms. So the plants can only absorb the nitrogen in the form of inorganic. So all the different forms should be converted into inorganic form in order to be absorbed by the plants, and also we and the animals absorb the nitrogen indirectly from the plants. Plants are the only living organisms which can directly absorb the nitrogen. So with the aid of microorganisms. So this is about nitrogen fixation. So this is why it is important to fix the nitrogen. It is just the conversion of unavailable form of nitrogen to the available form so that it can be utilized by the plants to start their metabolic activities. So this is about nitrogen fixation. Let us discuss about biological nitrogen fixation. So biological nitrogen fixation is a biosynthetic process. So what is biosynthetic process? So biosynthetic process means synthesis of a chemical compound by the living organisms, right? So what is biological nitrogen fixation then? So it is nothing but the reduction of nitrogen into the ammonia or ammonium form. So this is what biological nitrogen fixation means. So the atmospheric nitrogen is converted into either ammonia or ammonium form. So this is called as biological nitrogen fixation. So this is with the help of the bacteria called as nitrogen fixing bacteria. So we will discuss about these different classes of bacteria in detail in upcoming slides. The next process is ammonification. So what is this ammonification? So the ammonification is the conversion of organic form of nitrogen into inorganic form. As I told you, the inorganic form is the one which is directly utilized by the plants. So the ammonification helps in the conversion of the organic form into inorganic, nothing but the unavailable form of nitrogen to available form of nitrogen. So the organic form, what is the source of the organic form of nitrogen? It is the organic matter. From the organic matter, directly it will be converted into inorganic form with the through the process of ammonification. And in the soil, most of the nitrogen is present in organic forms. So, this should be converted into inorganic form to be effectively utilized by the plants. The next process is called as nitrification. So, what is nitrification? So, once the biological let us talk about BNF again, biological nitrogen fixation. So, once the nitrogen is converted into ammonium form or ammonia form, ammonium ion form, or from the soil the organic form is converted into inorganic form, which is again ammonium or ammonia. So, both of these should be converted into nitrate form to be utilized by plants. So, this process is called as nitrification. So, this is a series of events where the biological or the atmospheric nitrogen is first converted into ammonium or ammonia by the nitrogen fixing bacteria, or from the soil. If the source of the nitrogen is soil, from the soil the inorganic nitrogen is converted into organic nitrogen, which is by the process of ammonification, which can be then converted into nitrate, which is directly utilized by the plants. So, this process is called as nitrification. So, this nitrification is done in two-step process, where in the first step the ammonium ions are converted into nitrite. So, with the help of the different class of bacteria which are called as nitrosomonas and nitrosporia, thirdly it is nitrococcus. So, these are the three different classes of bacteria which will help in the conversion of ammonium ion to nitrite ion. So, this is the first step of nitrification. The second step of nitrification is conversion of nitrite to nitrate, and this is aided by the bacteria which are nitrosporia again, nitrobacteria, nitrobacter, and lastly it is nitrococcus. So, this is a nitrification is a two-step process where the ammonium ion is converted into nitrate with the intermediate nitrate form with the two different classes of bacteria, where majorly it is nitrobacter in case of conversion of nitrite to nitrate, and ammonium to nitrite it is nitrococcus. So, these are the two important bacteria that you should remember. And next process is called as denitrification. So, what is denitrification now? So, this is just the opposite process of nitrification, where the nitrate which is in the available form now is again converted into unavailable form, which is atmospheric nitrogen. So, this process is called as denitrification. So, this denitrification, one important thing to be remembered is it is in anaerobic conditions by anaerobic bacteria. So, what happens, this anaerobic bacteria in the absence of oxygen will utilize the nitrate as an electron acceptor during respiration process. So, which will reduce this nitrate into the atmospheric nitrogen. So, this is the reason why the nitrate is converting into nitrogen only in the anaerobic conditions. So, in the anaerobic conditions where there is no oxygen, the anaerobic bacteria will utilize this nitrate as an electron acceptor, and this will transfer this nitrate into nitrogen form. So, it will be unavailable form. So, that is why the process is called as denitrification, which is just the opposite of nitrification process. So, this is about the different transformation process that are occurring in the environment for conversion of different, like one form of nitrogen to another form. Okay. So, this was about different transformation process. Next, we shall discuss about the nitrogen fixing bacteria. Talking about the nitrogen fixing bacteria, they can be categorized into two different types. One is free living and second one is symbiotic form or symbiotic bacteria. So again, in the free living bacteria, we can just categorize them as aerobic and anaerobic. So aerobic bacteria and anaerobic bacteria. So the example for aerobic bacteria is Azospirillum and Azotobacter, and for anaerobic bacteria it is Rhodospirillum, and secondly it is Clostridium. So this Rhodospirillum is a photosynthetic bacteria, and secondly it is Clostridium is a non-photosynthetic bacteria. So this is about the free living bacteria which will fix the nitrogen. So talking about the symbiotic bacteria, we can again categorize this symbiotic bacteria into two different types. One is nodule forming bacteria, and secondly it is non-nodule forming bacteria. So nodule formation majorly takes place in case of legumes. It is majorly observed in case of legumes except rajma. Rajma is the exception. And secondly, it is also seen in some of the non-legumes which are ulnus, casuarina and myric. So these are the non-legumes in which the nodule formation takes place by the bacteria, which are the Frankia or Actinomycetes class. So these are from the Actinomycetes class which will form symbiotic relationship with these certain plants. So without these are the non-legumes. So again, coming back to the legumes, we know that all the major legumes or all legumes except rajma will form the nodules with the symbiotic bacteria for the effective biological nitrogen fixation. Like the examples of the bacteria are Rhizobium, Bradyrhizobium and Azorhizobium. And next we are talking about the non-nodule formation. It is majorly by Anabaena azole, which is a cyanobacterium which have the symbiotic association with water fern azole. So the name Anabaena azole. So it has a symbiotic relationship with water fern, which is Azolla. So this is about different categories of nitrogen fixing bacteria. So for the exam point of view, you have to remember these examples which are free living, which have symbiotic relationship, and which are nodule forming and which are non-nodule forming. And in the nodule forming, in the legumes, the nodules are majorly formed in the roots, but we have like stem nodulating bacteria are also there, where it is in Sesbania the nodules are formed in the stems, and for all other it is most commonly formed in the roots. So this is about the classification of bacteria or nitrogen fixing bacteria. Next we will start our discussion about nitrogen fixation and assimilation. So I will just give you a brief overview of how this occurs. So I told you there are two major sources of nitrogen. One is atmospheric nitrogen, secondly it is in the soil in the form of inorganic matter, right? So if soil organic matter, okay. So the atmospheric nitrogen is converted into ammonium form through biological nitrogen fixation. Secondly, the soil inorganic matter is converted into ammonium form through the help of ammonification. So this is the common form that is formed by biological nitrogen fixation and ammonification. So once this ammonium ion is formed, this is through the process of nitrification, it is converted into nitrate, which can be directly utilized by the plants. So if this is the plant roots, ground surface, and this is the roots, the plants can directly absorb this nitrate from the soil, or so the plants can directly absorb the nitrate in the nitrogen in the form of nitrate or directly in the form of ammonium ion. Okay, so this nitrate, once it is utilized by the plants or transferred into the roots, this transfer of nitrate into the roots is by the active process because of proton gradient. So what I mean by proton gradient, it is proton difference in the proton concentration, so which is generated by H plus ATPase. So because of this difference, there will be an uptake of nitrate by the plants. So once the plants have took this nitrate, so again this nitrate is converted into ammonium form and next to amino acid form. So this is how the nitrogen is utilized by the plants, or directly the ammonium form will be converted into, or it will be utilized by the plants and converted into amino acid form. Here the process is called as nitrification. This is ammonification, conversion of organic form of nitrogen to inorganic form, that is ammonium form, and from atmospheric nitrogen it is biological nitrogen fixation. So this is the complete overview of nitrogen fixation and assimilation. So assimilation is nothing but the utilization where, yes, like it is observed in the form of nitrate, but nitrate will be again converted into ammonium form to be effectively utilized by the plants. So this is called as assimilation. So broadly there are two different ways of how this fixation and assimilation takes place. So first method is by soil or soil nitrogen, and secondly it is biological nitrogen fixation of atmospheric nitrogen. In these two alternative ways, from the soil, so the nitrogen from inorganic matter is converted into organic form to inorganic form, that is ammonium form. Next there can be nitrification, which will convert it into nitrate. So once it is formed into nitrate, it will be absorbed by the plant roots. So in the roots there will be conversion of nitrate to ammonium form, which is utilized by the plants by converting it into amino acid. So now I have explained about nitrification in the earlier slide. So now we'll just talk about how this nitrate is again converted into ammonium form. Conversion of nitrate to ammonium form. So this is again a two-step process where in the first step nitrate is converted into nitrite, and in the second step it is nitrate which is converted into ammonium form. So the conversion of nitrate to nitrate is by the enzyme nitrate reductase, and secondly it is nitrite reductase. So many students will confuse in this particular enzyme aspects where it is nitrate reductase for conversion of nitrate to nitrate, and for conversion of nitrite to ammonium it is nitrate reductase. I hope this is clear. So again, going further, we can also discuss about the enzyme structure, the nitrate reductase. So this nitrate reductase is made up of two identical subunits. So each subunit is made up of three prosthetic groups. So prosthetic groups contains FAD, heme group, and also molybdenum containing organic molecule, so which is called as terran, TEPTERIN. So these are the three prosthetic groups which are present in the two identical proteins. So these are the part of nitrate reductase, and this requires a reducing agent for the conversion of nitrate to nitrate. So the reducing agent which is utilized is NADPH here. So this is a step 1. And in the second step, talking about nitrate reductase, the structure of nitrate reductase. So it is a monomeric protein. It's a monomeric protein with again two prosthetic groups. Earlier it was 3 here, it is 2 now. So two prosthetic groups are Fe4S4 and serum. This serum is nothing but a cyclic tetrapyrrole with one iron atom in the middle. So these are the two important two prosthetic groups which are present in the monomeric protein, which is nitrate reductase. And this is how the nitrate is converted into ammonium form again in the plants. So since the accumulation of the ammonium in the plants is toxic, it should be immediately converted into amino acids. So how this ammonium is converted into amino acid? So this is again by two-step process with the help of the enzymes, two enzymes which are glutamine, both are glutamine, but first one is glutamine synthetase and second one is glutamine synthase. So the ammonium is first converted into glutamate and then to other amino acid through the help of transamination reaction. So this is how the nitrogen is incorporated into the amino acid. So this was from the soil, from getting conversion from organic to inorganic. So inorganic through nitrate form, from nitrate to again ammonium form, from ammonium to amino acid form. So you have to remember the different steps and also different enzymes that are involved in this sequential steps. Okay. So this is about the first pathway where it is from the soil, where the source of nitrogen is from the soil. Secondly, we can start our discussion about biological nitrogen fixation, which is also very important. Biological nitrogen fixation. So as we know, in the atmosphere it is 78% of nitrogen is present. So this 78% of nitrogen in the atmosphere, it should be converted into available form to be effectively utilized by the plants, right? So this atmospheric nitrogen should be converted into either ammonium form or nitrate form again. So how this conversion takes place? So this conversion is again called as this nothing but biological nitrogen fixation, which I already told you earlier. So the biological nitrogen fixation can be again categorized into two different classes. One is natural process, where the biological nitrogen fixation occurs naturally, and second one is industrial process. So now let us stick our discussion with the natural process, under which we can again have two different steps. One is biological nitrogen fixation, second one is non-biological nitrogen fixation. So don't get confused, this is a nitrogen fixation. Let us consider this as just a nitrogen fixation. So the nitrogen fixation basically occurs in two different ways. One is natural fixation and second one is industrial process. Under the natural fixation, it is either through the biological nitrogen fixation with the help of organisms, microorganisms, that is why it is biological nitrogen fixation, and second one is non-biological. Non-biological examples can be lightning and also photochemical reactions. So now we have to discuss this biological nitrogen fixation. So the biological nitrogen fixation is through the prokaryotic organisms, and again it is a biosynthetic process which I told you earlier, and the prokaryotes which are capable of fixing this nitrogen or atmospheric nitrogen, they are also called as diazotropes. Just remember this term, diazotropes. This biological nitrogen fixation is conversion of nitrogen into ammonium form, where it generates one molecule of hydrogen with two molecules of ammonia for each molecule of nitrogen fixed, and it requires 8 electrons for this process. So this is the reaction, and it is catalyzed by an enzyme complex called as nitrogenase complex. See, this nitrogenase enzyme complex is again present in the three different forms based on whether they require molybdenum, vanadium or iron. So the most studied nitrogenase complex has molybdenum as a cofactor. Out of these three different types, most of them have the molybdenum as a cofactor. So talking about the structure of this nitrogenase enzyme, so this is again made up of two different proteins which are dinitrogenase, reductase, and secondly it is dinitrogenase. So first one is dinitrogenase reductase and secondly it is dinitrogenase. So this is also called as Fe protein, that iron protein, and secondly it is MOFE protein. First one, the dinitrogen reductase, it is a dimer and this is a tetramer. So at least you should be remembering these things, like at least about the subunits, the two proteins, and it is called as Fe protein, and secondly it is MOFE protein. Out of this second structure, second enzyme complex, or the nitrogenous enzyme complex, the second protein, which is dinitrogenase, this is the site for nitrogen fixation, where it is with the help of MOFE cofactor. This is the actual site of nitrogen fixation, where the fixation of nitrogen takes place. Out of these two proteins, it is in the dinitrogenase. And the production of this nitrogenase enzyme or synthesis of this nitrogenase enzyme are because of the enzymes which are, or because of the genes which are known as, popularly known as NIF genes. So NIF genes are those genes which will synthesize this nitrogenase and have the catalytic process and do the catalytic process of nitrogen fixation. So along with these NIF genes, there are also called as fix genes. So these are nothing but the accessory genes which will help in the regulation of the nitrogenase in the nitrogen fixing bacteria. So this is synthesized in bacteria, don't get confused. So bacteria will have the genes which are called as NIF genes, which will synthesize the nitrogenase enzymes required for nitrogen fixation. So talking about the process, the nitrogen, since biological nitrogen fixation is the reduction process, it requires electron donor for conversion of nitrogen to ammonia. So the electron donor is reduced ferredoxin. Just remember this compound, it is reduced ferredoxin, which will act as a electron donor for the conversion of atmospheric nitrogen into ammonia. And then talking about the process of this nitrogen fixation, the nitrogenase complex which is formed, this is sensitive to oxygen. So this particular complex is sensitive to oxygen, where it will be irreversibly inactivated in the presence of oxygen. So it is mandatory that this whole process should occur in anaerobic conditions. Since in the presence of oxygen it will be inactivated, this complete process of nitrogen fixation should be going on, or should be done in the anaerobic conditions. So if we talk about the bacteria, so for anaerobic bacteria there won't be any problem. In aerobic bacteria there won't be any problem, they can fix the atmospheric nitrogen, since they do not have, like, they will do it anaerobically, so it won't be causing any problem to them. So when we talk of facultative bacteria, which are purple photosynthetic bacteria, they will only fix the nitrogen when the anaerobic conditions are provided, otherwise they are not able to fix the nitrogen. So this is for facultative bacteria. So we have third category, which is anaerobic bacteria. So how does this anaerobic bacteria manage to fix the nitrogen, since it should be only fixed in anaerobic conditions? So there are some adaptation. So in case of cyanobacteria, there is a formation of a specialized structure called as atrocyst. In the atrocyst, anaerobic conditions will be provided, or anaerobic conditions will be created, which will help in the nitrogen fixation. So this is how the cyanobacteria have adapted themselves with the formation of specialized cells called as atrocysts, where the nitrogen fixation occurs. So in other cyanobacteria where the atrocyst is not forming, only in the anaerobic conditions there can be nitrogen fixation, otherwise they are not capable of nitrogen fixation. Then talking about fourth category, that is symbiotic bacteria, how do they manage to fix the nitrogen? So in the symbiotic bacteria, which are mainly rhizobium, we can take examples. So this will fix the nitrogen with the help by maintaining very low concentration of oxygen. So how do they maintain very low concentration of oxygen? With the help of a compound called as leg hemoglobin. By producing this leg hemoglobin, which is a homologue of hemoglobin, so which gives the pink pigment, pink color to the legumes, nitrogen fixation is done. In the symbiotic bacteria, the anaerobic conditions are provided in such a way that by producing the leg hemoglobin, which will give a pink color to the legumes, it will fix the atmospheric nitrogen. So where this leg hemoglobin is called as oxygen scavenger, which will maintain very low oxygen concentration in the cell. So this is all about the nitrogen fixation, how it occurs, the general overview of the complete process of ammonification, nitrification, denitrification, and also biological nitrogen fixation. I hope like you got very clear about the different enzymes which are involved in the process at what stages. So if you still have any doubts, you can also comment on the video, or you can directly contact us through the number we have provided. So if you just like the video, if you just like the content, please subscribe our channel and also share it with your friends. So if you just like this content, just comment on this video that if you want the next video where I will be explaining the nodule formation, like how actually the nodules are formed in the rhizobium for the biological nitrogen fixation, I can make another video for that. So if you need that video, you can just comment on the video. Okay, so that will help us to that will give us the encouragement to make more such content. Thank you.